So, I am very happy to welcome you to this webinar, where we will be going into detail on the new substances on our SIN list, the list of substances to prioritize for phase out. So, these new substances were added on the 14th of October at our event in Brussels. And I can see that many of the people that attended the event are also here today wanting to know more. So, welcome to you. But also welcome to all the other people who are new to this update. And with me today, I have both our in-house senior toxicologist, the person behind this work, Dr. 𝘁𝗵𝗶𝗀, but also researchers that have been involved in finding and evaluating the substances. And that is Dr. Agathe Boer, ecotoxicologist at Good Point, and Professor 𝘁𝗵𝗶𝗀𝗃 at the Norwegian University of Science and Technology. And I am Theresa Kjell, Head of Policy at ChemSec, and I will guide you through this webinar. And the recordings and the slides from today's webinar will be made available. And after the presentation, there will be an opportunity to ask questions to the panel. So please note down any questions that you have in the Q&A box below in Zoom, and we will do our very best to respond to them. So first out today is Dr. 𝘁𝗵𝗶𝗀𝗃. She is a senior toxicologist at ChemSec and responsible for the SIN list. Hannah is an ecotoxicologist by training and holds a PhD in environmental systems analysis. She will now introduce the SIN list and its updated content. The floor is yours, Hannah.
Thank you, Theresa. Thank you for that nice introduction. So I will give you a brief overview here, starting this webinar. First, to give you an overview of the content of the launch that we did now in 2025, and also briefly relate this to the SIN list as a whole. And then finally show you on the SIN list where you can find these new chemicals and information about them. But first, very, very short, I'm sure many of you are already SIN list users, but for those who are not, I would like to tell you that the SIN list is a globally used database of chemicals that are likely to be banned or restricted in the future. And SIN is an abbreviation for substitute it now. So as Theresa said, it's our list of chemicals to phase out. And the chemicals that we put on the SIN list, those are chemicals that ChemSec and the experts that we collaborate with have assessed to meet the REACH criteria to be substances of very high concern. So as of today, we have 1,473 entries on the SIN list, and 25 of these were added in October 2025. So we'd like to think that the SIN list is one of the most progressive chemical standards in the world. And we've shown many times that we are before regulation, and we intend to keep being so. Next slide, please. So the SIN list is now quite a mature tool. It was launched in 2008. And since then, we've added endocrine disrupting chemicals and persistent bioaccumulative and toxic chemicals. In 2019, for the first time, the persistent mobile and toxic chemicals were added together with carbon nanotubes. And the last update that we did by ChemSec initiative was in 2023, when 370 PFAS perfluorinated alkylated substances were added. And then click, Jonathan. No. And now in 2025, we are adding persistent mobile and toxic chemicals again. And for the first time, neurotoxicants. And this is what we will learn more about in this webinar. Next slide, please. So we're using the SIN list then. We have 37,300 persons visiting the website 135,000 times during the last year. And of these, 13,000 were registered users. And these people come from all over the world. They come from the US, Sweden, Germany, France, and also from countries in Asia, for example. Most of these are professional users who are active within chemical industry and in manufacturing, downstream chemical users within manufacturing. And this is also the intended audience for the SIN list. And when we asked these users what they use the SYN list for, they also said that they use it as we intended to be used. So they use it to see what can be expected to be banned and also as a phase-out list. Next slide, please. And we were very happy because when we asked our users how useful the SYN list is to them, more than 80% said that it was very useful or useful. So that's very good. Next slide, please. So the SYN list is one of several tools for chemical management that we provide at ChemSec. They are all online tools and they are free of charge. And I would like to highlight another example, and that is our marketplace tool. So Marketplace is a business-to-business platform where we provide safer alternatives to hazardous chemicals. And today, Marketplace has almost 800 alternatives on the website, and it's continuously growing. So now when we have added new SYN list chemicals to the SYN list, chemicals to be phased out, we will also, of course, go and look for safer alternatives for these chemicals and populate Marketplace. Next slide, please. So to the update that we did now, first I would talk about the persistent and mobile chemicals. So of the 21 new entries, 18 are persistent and mobile chemicals. It's a heterogeneous group of chemicals, but they have in common that they are all fairly small molecules with heteroatoms. So they are polar, meaning that they're water soluble and they can move through soils and contaminate our water resources. And of the 18 entries, 9 are persistent, mobile, and toxic. 13 are very persistent and very mobile. And to make the math go together, you see that 4 are very persistent, very mobile, and also toxic. To find these 18 chemicals for the SYN list, we started from a list of several hundreds of chemicals that are persistent and mobile. And then we prioritized to a short list based on urgency of action. So we looked at use amount and likelihood of emission, as well as if these chemicals are found in the environment already. And then when we had a short list based on this urgency of action, we looked at the data availability. So the chemicals from this short list that were not on the SYN list now, they're not deselected, most of them because they're not persistent or mobile. They are just deselected because we don't have enough evidence at this point in time and hence should be studied further to see what their properties are. We also made use of some predictive tools to see which ones of these hundreds of chemicals are likely to meet the persistency and mobility criteria. But in the final assessment, we gave the most weight to experimental data. Next slide, please. So if you want to find these chemicals on the SYN list, you can make use of the filters that we have. You see them on the left-hand side. So if you enter the SYN list, the starting point is these 1,473 chemicals, and then you can use the filters. So here you have the health and environmental concern. If you click the PMT filter, you see that we have 53 such entries. And then if you click the filter for the October 2025 update, you see that we have 18 such entries. And I will show you some groups and some chemicals within these 18 entries. And next slide, please. So one way to look at these is to use the SYN groups. That's another filter you have on the left-hand side. And you can see here, for example, that 13 of the 18 entries are nitrogen compounds. So we have several groups here represented: aromatic amines, aminocarbonyl compounds, nitro compounds, amines, triazines, and benzotriazoles, for example. Next slide, please. So if we look into that a bit more and exemplifying with triazoles and triazines, why do I choose them? Yeah, well, because in the literature, we often see PFAS, triazines, and triazoles pointed out as groups that include many persistent and mobile chemicals. And if you remember that we put 370 PFAS on the SYN list in 2023, none of them are, of course, included in this update. But we do have triazines and triazoles. So we have 2 benzotriazoles included in this update. And that SYN group did actually exist before October 2025 because we had a number of persistent and bioaccumulative benzotriazoles, some UV filters. And now we include some benzotriazoles that are smaller molecules and then are persistent and mobile. So they are used, for example, as corrosion inhibitors. For this new update of the SYN list in 2025, we also added a new SYN group. So triazines is a new group on the SYN list. And we have 5 entries that are triazines. You see some example structures here on the slide. And they are used for many, many different things. For example, as water treatment chemicals, as stabilizers, processing aids, etc. And we have both consumer uses and industrial uses represented for these chemicals. Next slide, please. Another interesting group is the halogenated compounds. So we have 4 such entries in this new update. So you see we have some overlap between the groups. So cyanuric chloride was included also in the triazine group. But here we have another interesting example, which is the 3-chloroacetic acid, so the TCA. And it's a cousin, you can say, to 3-fluoroacetic acid that was already on the SYN list since 2019. And TCA is sometimes used to replace TFA, for example, in chromatography. And here we can show then that that substitution is not to be recommended, and one should look for yet safer alternatives. Next slide, please. So that was it on the persistent and mobile chemicals. And then we also added the neurotoxicants. For the first time, we add chemicals to the SYN list purely because of their neurotoxic properties. And we have 3 entries that we classify as neurotoxicants, or that we mark as neurotoxicants. And we selected these from the classification, labeling, and packaging regulations harmonized classification, where we looked into the chemicals that have a category 1 classification for specific target organ toxicity, where effects on the nervous system was indicated. And for these, so there are 30-something chemicals with that classification. We looked at the chemicals that were most potent and toxic at the lowest exposures, with very severe effects and also irreversible damage. And where data were available to clearly support equivalent level of concern, and hence that they are meet the criteria to be substances of very high concern. Next slide, please. So these 3, you find them as well on the SYN list. If you use the filters to the left-hand side, you can click the neurotoxic filter under health and environmental concern. And you see that 2 of these are tin compounds, and one is a pyrithion. And they are used for different things, for example, in plastic manufacture and in glass, and also in sanitation. And you will learn more about the health hazards in a bit when Agathe is giving her presentation. Next slide, please. So that was a brief overview. You can find much more information on the SYN list, both on the actual chemicals and also the method that we've used to select them and assess them. So please use these QR codes or click your way through via chemsec.org. Next slide, please. And finally, I would just like to acknowledge all the people who've been involved in this work. So firstly, the PMT update is done by ChemSec in collaboration with researchers in the EU Horizon project 𝘁𝗵𝗶𝘀. And the neurotoxicity update was made by ChemSec with our expert consultants. On top of that, we've had external reviews by independent academic researchers, as well as the advisory committee for the SYN list. So with our warmest thank you to you all to make this possible. Thank you.
Thank you so much, Hannah. And I would also try to remind you now to post questions in the Q&A box. Don't hold them in, and we will try to answer them at the end. So with that, we're moving on to the next speaker, and that's Professor Hans-Peter Arp. He is an expert at the Norwegian Geotechnical Institute, a professor at the Norwegian University of Science and Technology, and coordinator of the research project 𝘁𝗵𝗶𝘀, among other things. Hans-Peter has been involved in each step of the selection of the PMT substances, which we will hear more about now. The floor is yours.
No, thanks very much, Teresa, and also thanks to you, Hannah, for that great introduction. So for my part of this webinar, I will be going over some concepts that Hannah already brought up, but putting some more background in terms of the science and regulatory context towards these new substances on the SIN list, particularly the PMT, vPvM ones. The next slide, please. So where PMT and vPvM come from are this new hazard class that was introduced into the CLP regulation of the European Union, or the chemicals labeling and packaging regulation, alongside the PBT, vPvB hazard classes and endocrine disruptors. And this hazard is defined in the regulation as, for PMT, can cause long-lasting and diffuse contamination of water resources, and for vPvM can cause very long-lasting and diffuse contamination of water resources. And the introduction of these hazard classes was something that was brought forth as part of the EU Chemical Strategy for Sustainability, which is also related to the Green Deal. And since the publication by the Commission of these new hazard classes, there's also some guidance that was made by the European Chemicals Agency, which gave guidance about how to assess chemicals if they meet this criteria or not. And I want to emphasize what this is related to, why it's important that we have these labels on chemicals and new chemicals and old chemicals on the market, is to see risks that can happen early on before they happen, and to take actions on that before risks are manifested. And this is part of the Commission's generic approach to risk assessment management, that when there are certain hazard classes that are evident, there'd be an automatic trigger of certain measures. And this can include developing ways of reducing exposure, reducing emissions, already setting up monitoring, or communicating in your supply chain. It can have various effects, and of course, a substitution to safer if you're still at the design stage. So that's the background of these hazard classes. I'll now go into the workflow of how we decided on, or how we came to the conclusion of adding these new 18 substances. Next slide, please. So what we did, this was a work that was done, it was a collaboration with the 𝘁𝗵𝗶𝗀 project and ChemSec as part of our market transition work package. And within 𝘁𝗵𝗶𝗀, we had a subgroup of various experts, and this was established in March 2024. And these were experts from NGI, from 𝘄𝗲𝗯𝗶𝘀 in Germany, from University of Luxembourg, from 𝘃𝗃 in Switzerland, 𝘹𝗲𝗃 Stockholm University, and the German Environmental Agency, 𝗹𝗲𝗿𝗃. And we all had different expertise, and we're contributing to this in different ways based on our expertise. Next slide, please. Or yes, an animation, I should say. So we did start with this document you might see in the corner, this UBA document. So this was something that I was involved in previously to try to find potential PMT, vPvM substances on the REACH, that were registered under REACH at the time in 2019. So we had this as a starting list. But the very first thing we had to do with that starting list was to remove the substances that were already on the SIN list, because that was decided that we will not add PMT classifications to those. So there was 93 substances that we removed that were already prioritized on that initial UBA list. And these were not necessarily PMTs. They could also have been carcinogenic, or we lost a lot of PFAS substances due to the large PFAS addition. Then we had an exercise to discuss how we were going to prioritize, because we couldn't do deep dives into all these chemicals. So we first wanted to have a way of prioritizing, and we discussed about the high tonnages, the regulatory status. For instance, if there's already a lot of regulatory action on a substance, it was less interesting for inclusion. We had a 𝘁𝗶𝗻 approach to substance grouping, which is why we've had these focus on triazenes and triazoles, because we're collecting data on them specifically, monitoring data. So all these were done to make an initial short list. And next animation. Then we actually refined it further, really based on data availability, because we needed to have enough evidence to feel it was justified to add these to the list. And then finally, when we had our very short list, we had some external discussions with people that weren't involved in the work, such as an external scientist, Professor Martin Schenger, and also with the SIN list advisory committee. Next slide. So I'm going to go a bit more into this weight of evidence approach, because this is what a lot of the decisions were based on. And And this is really about how much data is available. There is guidance. We did use the 𝘂𝗍's guidance for the CLP to formulate this weight of evidence approach, so it was based on this. But what we could find with just based on the available data, that the assessment of mobility, the M and the VM, and the toxicity was relatively straightforward. For toxicity, we would just have to look if there was any harmonized information available, or if our experts, our toxicological experts, would go into the data and say there was enough reason to conclude a particular toxic category. For mobility, it was similar, and mobility is defined based on the log KOC value, and this is a log value. And so you'll find that some of the substances had log KOC values which are likely very far below the threshold. So it wasn't a question if they were mobile or very mobile or not. And maybe for ones that were near the threshold, we had to have a little bit more data. But what was most difficult in all this work was the persistency assessment. That is where we had the least data available. And so we spent actually most of our time looking at persistence information. Next slide, please. And I'm just going to go a little bit in more detail into persistence, because I said this was what was making or breaking if a substance would make the list or not. I'm not going to go through all this information. I'll just give you a general overview of how it works. So from the CLP, and also this is also the REACH and the biocide regulation, there is a definition for persistent or very persistent, and it's based on half-lives in specific media, such as fresh water. A substance is considered persistent if it has a half-life of about 40 days, and very persistent of 60 days. And then on the right of your screen, we're going into how to assess that. And there's a lot of lot of evidence, or weight of evidence, is put on these simulation tests, which are just simulating the half-lives in the laboratory. But if this is not available, then one can use other types of information like ready biodegradation tests, or models, or QSARs, or inherent biodegradability tests. And there's different ways of weighing this evidence. And this is a bit complicated to kind of explain, but I'll just show you how this could work in principle. So next slide, please. Yes. So when we did look at this, we did have to look at all available lines of data for persistence. So if we took a simulation test or ready biodegradable test, we took everything that we could, for instance, find in the REACH dossiers, but also went into the literature. And next bullet point. And a key thing that we had to do when we looked at this data is see if they were compliant with the recommendations and the guideline. Now, something that we would find often with a lot of the data, such as that was reported on REACH, was they would use a very old test method that wasn't compliant with the newest OECD guidelines, for instance, and therefore that type of data would be inadmissible. And it does take a little bit of expertise to recognize if the data is admissible or not. Next slide, or next animation. So we did assign certain types of weights of evidence based on the type of data, whether it would be low, medium, or high. Next bullet point. And here you see it here, at least for persistency testing. So a high weight of evidence would be related to a simulation test or of the degradation half-life in the lab, according to compliant testing. We had a medium weight of evidence for good quality, compliant, readily or inherent biodegradability tests and mesocosms, perhaps giving it a bit more weight of evidence that is typically used by 𝘂𝗍. And the lowest we would put for monitoring data or QSAR data, because this is quite unreliable for determining media-specific half-lives under standard conditions. Next slide, yeah. And so, yeah, so basically a chemical was considered to meet the P or VP criteria if it was expected that the half-life exceeded the threshold that I showed earlier. So let's go to an example. So I'll go to the example of a cyanuric acid, which is one of our new substances. It was very persistent and very mobile. And it's also of concern because when it's present together with melamine, it can lead to kidney stone formation. So it's considered nephrotoxic. And we're seeing in a lot more drinking water resources that melamine and cyanuric acid are going together. So we concluded it was vPvM. Next slide. But this might seem to some they might have thought it was not persistent. So when I look actually at the REACH dossier for a cyanuric acid, instead on the lead dossier, which is combining all the information in the other dossiers, that this substance is not persistent. And this was based on rapidly biodegradable tests. So then why were we disagreeing with this dossier and saying it was persistent? Next slide. It was because that the readily biodegradable tests they were basing this on were non-compliant. These were old tests from the 70s, and they're not compliant with the new guidelines, such as OECD 301 or 302. So the tests did not resemble conditions, and also the data did not report the test conditions. And so there was other evidence why we did not include it. But meanwhile, in the same dossiers, they also included a aerobic biodegradation test in marine water after OECD 306. And this was done and described in such a way that it resembled a simulation test. And this was a very good quality test with a lot of the data provided. And they reported that they had a 4% degradation of cyanuric acid after 60 days, which is more than sufficient to conclude that this is P and VP. And so again, this OECD 306 test was considered high weight of evidence, unlike the low or actually non-compliant data that was used. So that is why we concluded it to be persistent. And I'll just mention before I talk about this slide that you also saw cyanuric chloride on there. And that what that does in the environment is that it loses its chlorine right away, and then it transforms into cyanuric acid. So the evaluation for cyanuric chloride was similar to cyanuric acid. Now we can go to the next slide. And this is another example of our grouping approach with the triazenes and the triazoles. So this is looking at benzotriazole and the 4,5-methylbenzotriazole. So that's 3 different substances. But there was a lot more data for the benzotriazole with high weight of evidence that we could find that it was very persistent in water, very persistent in soil. It had a mobility below the cutoff with OECD tests. It's known to be chronic toxicity to aquatic organisms, but our deep dive into the toxicity literature also helped us find it was teratogenic to mammals. And there was less data for the 4 to 5-methyl-1-benzotriazoles, but there was some to indicate persistence and mobility, and using read across through their structural similarity, we're also concluding that it is toxic as well. So substituting one for the other would be an example of regrettable substitution. And also these can be degradation products of other substances. So by proxy, substances that degrade to these 2 substances should be considered PMT, vPvM. So that is some context. And then I'll just want to say thank you to everyone that helped with this work. So my next slide. So this is the 0PM subgroup that helped us make this list. And of course, I'll also acknowledge funding from the European Commission for 0PM. And thank you for your attention.
Thank you so much. And again, I would like to encourage you. I see some have posed questions in the Q&A already, but please feel free to do that, and we will answer as many as we can. So with that, I want to bring us to the next speaker, Dr. Agathe Boer, a senior advisor at GoodPoint. Agathe has a background in ecotoxicology and environmental sciences, with experience from working on a range of different pollutants, including nanomaterials, microplastics, and plastics additives. She has assessed the neurotoxic substances added to the SYN list. The floor is yours, Agathe.
Thank you, Teresa. I will share my screen now. Thank you for the nice introduction. Hello, everybody. I'm very happy to be here today and to present this work on neurotoxicants. But before we start on neurotoxicants, I would just like to say a few words about GoodPoint. So we are a consultancy company that focuses on sustainable business development, and we offer expertise that is both broad and in-depth, so covering all the relevant topics on sustainability, but also the entire value chain, and we are highly specialized. We offer expertise in ESG and sustainability reporting, climate and biodiversity, business ethics and human rights, management and quality assurance, and of course chemicals. And within chemicals, we have experts in life cycle analysis and environmental product declaration. We offer regulatory support, chemical management, so inventories, risk assessment, substitution, compliance, and of course, expertise in ecotoxicology and toxicology, which brings me directly to our neurotoxicants. So Hannah already introduced you the 3 substances that have been added to the SIN list. And now I'm going to present the method behind this update and give you some more detail on the chemicals. But I would also like to discuss why neurotoxicants are so relevant for the SIN list. And for that, I think it's important to understand what they are and how they can affect us. So the nerve system is highly complex and very finely regulated, with like all our nerves communicate between them and to all our organs, our muscles, even the skin. Everything in our body is connected to the nerve system. And this fine communication is ensured by molecules that ensure like good signal transmission, reception, fine modulation. So if anything like a chemical comes and disturbs this signal transmission, it can have bad consequences. And that's what we call here molecular effects, or effects at the molecular level. But neurotoxicants can also affect directly the nerve, for instance, inducing nerve death, or even directly damage the brain tissue. And these effects can be seen with the naked eye, and then that's what we call the clinical effects. And there we can have, for instance, uncontrolled movements, confusion, memory defects, personality changes, even psychotic behaviors. So we have different levels of impact, but how do we capture this impact? How do we measure? So for the molecular effects, that's usually in vitro testing. So it means we test directly on cell lines, cells, and that's usually short-term impact. For the physiological or morphological effects, that's seen following in vivo testing, so that's animal data, but that can also be seen in humans. So we can see, for instance, brain damage, and that's usually effects on the short to medium term. But then when it comes to clinical effects, that can also be seen following in vivo testing, and we have human data, and there we make a difference between clinical cases, which is usually a small group of individuals, and epidemiology studies. We can be up to hundreds of patients exposed in the same conditions. And that's very interesting for us because it's also longer term exposure usually, so months up to years of exposure. But why is this relevant? It's because for us, it's very important to understand the data we have and how it was obtained, because it's going to impact the way we assess the importance of this data. So we're not going to give the same weight to only in vitro data if we have in vivo data. And in the same way, only in vivo data on rats is not going to have the same weight as if we have data on hundreds of humans. So again, that's very important for us and for our assessment. And I'm going to come back to that soon. But now, a few words about neurotoxicants in general. So it's estimated that there are hundreds of neurotoxicants on the market, and depending on the sources, more than thousands. But it's only an estimation because today it's quite difficult to clearly identify neurotoxicants. They can be organic solvents, heavy metals, other industrial chemicals, and they can have potentially very severe effects and even suicidal effects. They can affect us at low level of exposure, and we expose usually chronically, so maybe over a period of years or even a lifetime. Now, when it comes to regulation and classification, for this reason, neurotoxicants are recognized as most harmful substances, and yet they do not have a harmonized hazard classification under CLP. Instead, they are covered under the specific target organ toxicity classification, or STOT, the target organ being, of course, the nerve system. But this STOT classification covers a wide range of effects, from very mild to very severe effects. So from there, how do we isolate and regulate the most harmful substances? So I'm sure you're all familiar with substances of very high concern, SVSC, that covers substances with the CMR classification, so carcinogenic, mutagenic, reprotoxic, but also PBT, vPvB, but not STOT. Instead, there's another classification that's interesting for us. That's the equivalent level of concern. And that is made for chemicals that are not CMR or PBT, but can still have effects that can be comparable to the effects of carcinogens, for instance, in terms of health effects and other factors that I'm not going to detail here, but in terms of severity, irreversibility, and delay of effects. So our objective here was to analyze the data on neurotoxicants and see if this toxicity was comparable to the effects of carcinogens. So how did we do it? So first, we selected substances that were classified as STOT, the CLP. And that's because it means that the substances already have been assessed by ECHA. So that gives us a guarantee on the severity and potency of effects. And then under STOT, it can be following repeated exposure or single exposure. We selected both, and with different hazard categories. And we selected the hazard category 1, which is the highest level of hazard. STOT can be about any organs. Of course, we only took what's related to the nervous system, and we also refined the results so there would be no redundancy with the same list, but also no chemical with same CMR, PBT, vPvM classification. Otherwise, we would lose the equivalent level of concern relevance. With that, we obtained 30 substances, for which we did an in-depth data analysis based on data we had from the ECHA databases. So that can be reports from the risk assessment committee, that can be rich dossier. And we assessed data availability, data quality. So that can be if the right studies have been performed in the right conditions, with the good controls, with the conclusions are supported by the results, for instance. We assessed data robustness, if there was enough studies and on groups big enough. And when all these criteria were met, we focus a bit more on the type of data we have, as explained before, in vivo, in vitro, animal, human, and on health effects. And now I'm going to say a few more words about the health effects. So when it comes to potency, basically, when we have effects at low doses, it means that we have high potency and vice versa. And for that, we use specific toxicity metrics. I'm not going to detail now. About the irreversibility, so some effects, some changes, intellectual alterations are known to be irreversible. Unfortunately, there's no general rule, and it had to be a case-by-case evaluation of the effect. It's similar with the severity. So some effects are known to be more severe than others. Here you have like some class of effects with a gradient of severity. And very quickly, a very short list of examples of effects that are considered severe and very often irreversible. So for instance, chronic brain damage, dementia, psychotic disorders, spinal cord disease. Those are the severe effects, among others. So when we assess the health effect for equivalent level of concern, we always have to consider together the potency, the severity, the irreversibility, and the delay of effect. But we give more weight to severity and irreversibility. And we also always have to keep in mind the type of data we have. So again, giving more weight to in vivo data, human data, epidemiology data, and keep in mind the data robustness. So following this data, this in-depth data analysis and assessment, we selected 3 neurotoxicants that were deemed to be of equivalent level of concern. And that's the chemicals I'm going to present a bit more now. So first, dimethyltin dichloride. So this chemical is used in the industry for the manufacture of chemicals and mineral products. It's used, for example, to stabilize PVC piping for glass durability, and it's also a catalyst in electrocoated paints. When it comes to neurotoxic effects, we have both animal and human data, which is very good for us. And the data shows clear signs of neuropathological lesions, clinical signs of neuropathology, developmental effects also. So in that case, it means that the female rats were exposed to the substance, and we observe the effects on the pups after birth and at adult age. When it comes to the human data, we have 2 clinical cases, so 2 groups of patients. In the first group, the patient showed memory defect, vigilance loss, insomnia, anorexia, disorientation, even epileptic seizures, but all the patients recovered in that group. In the second group, the patient showed similar effects, but also personality changes with aggressiveness, psychotic behavior, and even memory loss for more than 6 months. And all the neurological effects were seen up to 6 years after the exposure stopped. So here, we clearly have severe effects with lesions in the brain and in the spinal cord, altered postures in the rat, psychotic behavior, epileptic seizures. We have irreversible effects with, again, the lesions in the brain and spinal cord, neuron death, developmental effects. Even in the epileptic seizures, if the patient's recovered, the seizures can still lead to permanent damage. And when we have effects over 6 years, even if it's not a proof of irreversibility, it's still a very long term for effects. One thing that I did not mention is that these effects, they happen at quite low level of exposure. So here it shows that we have high potency. So for dimethyltine dichloride, we have a chemical that shows severe effects, irreversible effects, and has high potency. So clearly, dimethyltine dichloride is of equivalent level of concern. Now, methyltene-mercaptide. So this chemical is also used in the industry, but for the manufacture of plastic products. It's a stabilizing agent and a processing aid, and it has applications, for instance, in construction and building. An interesting fact is that it's quickly metabolized or quickly hydrolyzed in the body and releases DMTC, so the chemical I just presented. So the neurotoxicity assessment from ECHA is actually based on the assessment from DMTC. So here for methyltene-mercaptide, we have the same conclusions on neurotoxicity as for the previous chemical. And finally, for sodium pyrithione, this chemical is a biocide used both in industry and for consumer products. So in industry, it's used in functional fluids like cooling fluids, hydraulic fluids. It's used in lubricant, anti-fouling paints. And it's found in consumer goods like inks, cleaning products, and detergents. But it's also used to make fine chemistry, so make laboratory chemicals. So here we see that we have a much broader range of use, and this chemical is also used, produced in much higher volumes. When it comes to the neurotoxic effects, we only have animal data, but it's very robust. So it means that we have a lot of studies, and all these studies show consistently the same effects. So clear signs of neuropathy, muscle atrophy, nerve degeneration, retinal atrophy. The numbers I have here is just to show that this effect occur at very low concentrations. So here we have high potency. And interestingly, the females were 10 times more sensitive than the males. So here again, we have severe effects with limb paralysis, for instance, nerve degeneration, irreversible effects, again with nerve degeneration in the spinal cord, retinal atrophy, and high potency. So again, sodium pyrithione is clearly of equivalent level of concern. To quickly conclude, we have 3 neurotoxicants that show severe effects, high potency, irreversibility of effects, and we have robust data, including human data. So this data shows that health effects are of equivalent level of concern. So these 3 neurotoxicants are clearly relevant to the SIN list. And on that, I would like to thank you for your attention and be happy to take questions.
Thank you so much, Agathe, and thank you to all the speakers. We now have about 15 minutes for some Q&A. So great that you're putting on your camera. See if Hannah can put on her camera as well. We have received a few questions. So I would start with one on neurotoxicants to follow up on your speech. And I think, Agathe, you answered it to some extent, but in more detail, maybe we can start with Hannah to explain, out of hundreds of neurotoxicants, only 3 were selected. Then how can you say that neurotoxicants are so relevant for equivalent level of concern in the SIN list? Hannah, would you be able to answer that?
Yeah, I can start, and then I think Agathe would like to add to my answer maybe.
Great.
But as I mentioned already in my introduction, we started from the harmonized classification. So that's already, the chemicals that have been selected because they are highly hazardous and been in-depth assessed. And then we looked at the ones indicated to be effective on the nervous system, and then we picked the worst from that list. So that's one relevant starting point. And then we can imagine that there is many, many more chemicals out there that are not on the harmonized classification list, but that are potent neurotoxicants. And we have done some tries to see how can we start from other lists of chemicals or other information. And we think the there is a potential to do so, but we haven't finished that work yet. One thing that is limiting this is, of course, that this type of effect hasn't been in focus that much. So of course the ready-to-use databases, etc., are not in place. But another thing is that many of the most serious effects are those that are delayed, like what Agathe was just explaining. And then of course the standardized testing that we get to do under REACH, for example, it doesn't capture these effects necessarily. So the ALS concerns or the Alzheimer's concerns, etc. So that is another reason that for now, maybe the testing that we do on the many chemicals don't capture what we are worried about. So we need more research to make sure that we test for these relevant endpoints as well. And Agathe, maybe you want to add to this.
Yeah, exactly. You explained that very well. And I think there's a lot of data on neurotoxicants out in the scientific literature, but what's out there is not necessarily ready to use also for regulation and classification, because it doesn't capture the data that is considered robust enough, like human data, for instance. And I think this data is missing, like it's a vicious circle. So this data is not mandatory for the neurotoxicants, so the data is not produced. But if it was mandatory, then we would have the data that would help regulating. So yeah, the data that would be helpful for regulation is lacking a bit at the moment.
Yeah, the lack of data is a constant problem. So for the next question, it's one about half-life. It says here, one person asking about you refer to degradation periods of 40 to 60 days. It doesn't sound very persistent to me, the author is writing. Please, could you educate me on what half-life is, why a period of a month or so for degradation is considered P or VP, and what it means for human and wildlife exposure? Hans-Peter, maybe you could take this one.
Yeah, I could take a crack at it. So a half-life for chemicals is similar to what could be also considered with the stability of radioactive nucleotides. It's about the how long it takes for half of that substance to be disappeared from the surroundings. And so it's sometimes called the DT50 value. And it's specifically referring to the parent product. So it's not complete mineralization in this context. It's just the parent substance that we're looking at. And why these 40 to 60 days? This is probably the oldest, one of the older criterias, because this was originally developed 70s, 80s, 90s with the chemicals that were known to be persistent in the environment and causing problems with chronic aquatic toxicity, which means that if fish were exposed to them for a certain amount of time, they could have certain chronic effects that were not seen at a short time scale. From that, and this was way before my time when they came up with this half-life, they thought this was a reasonable amount of chemicals that were that stable in water. Were of a concern to look at if there's other avenues of chronic toxicity to fish, but also through the food chain, because if they're persistent and bioaccumulative, with that cutoff, they could also find a way to enter the diet because the fish would have that much time to be exposed to them. So that's like specifically for water, but then later this was extended to other media like sediments and soils. And there was a lot of discussions at various meetings, and then eventually for regulations like the Stockholm Convention on POPs, they made some decisions. So they used a 60-day criteria for water and similar 180 for soil. This was also taken up by REACH, but you saw a lot of debates and discussions in the 90s and the aughts. But and now we just stuck on those, and now we have to be consistent because with what is on the books for now. So that's the origin. Yeah. Thank you,
Hans-Peter. So now a question about the regulatory data. Is there any other authority providing regulatory data in the same level as ECHA is providing, or any other public source which is available? Maybe Hanna, could you start out with answering that one?
Well, I can just say that for the assessment that we did now, for the PMT assessment, we mainly use the data made available via the REACH registration dossiers, but we complemented that quite often from the Japanese database, so where they had done GHS assessments, a globally harmonized system assessment, which is to a large extent similar to the CLP. So for example, for toxicity, we use data from the Japanese databases. We've also used some American data. So the US authorities had evaluated, for example, persistency, and then we gave that a fairly high weight in our weight of evidence assessment.
Great, thank you so much. I have a question. It says, this update includes a focus on triazoles and triazines as PMT and vPvM substance groups. Are there other substances that belong to this group that may be considered as PMT and vPvMs? Maybe Hans-Peter, could you elaborate on that?
Yeah, sure. Yeah, well, the triazines and triazoles, these are actually very large groups. So we did a survey like how many triazines and triazoles are actually out there. And if you look at resources like PubChem, it's a crazy high number in the hundreds of thousands. Not all of them are in the global chemical inventory or produced at large scale, but there is quite a large amount. And you even see with the triazoles, some of them are even PBTs because they're associated with bigger molecules. But it does seem to me to indicate that these structures could be associated with a persistency hazard, but this would just be a first indication for me. This would have to be followed up by more data, because I think it's only certain types of triazines and triazoles that are of the most concern. For instance, if it's a hydroxybenzotriazole, it would probably degrade fast. So it depends what other bonds are to it. So I would say this is a mega group, but we probably have to define a more narrow scope of which triazines and which triazoles are hazardous. But I do think, yes, there are other triazines and triazoles that are hazardous that we have not identified yet, and we will look out for those.
Thank you for clarifying that. And the next question I have here is a bit of $1 million question. Hello, what are the next updates you plan based on the new toxicity ecotox properties, or more on looking for new and more data for substances to classify them under already listed ecotox properties? Hanna, this one is definitely for you. You feel okay to answer this one?
Yeah, it's a brilliant question. I mean, this is what keeps us busy, right, at ChemSec and in the SynList group. So there is, I can only give the general answer, but I think it's 3 strands of work. So of course, we're always on the lookout for, the next relevant hazard categories, like we're putting the neurotoxicants on the SynList now, and trust on us of being this watchdog looking out for the new hazards, and keep us informed if you feel that we've missed anything. In parallel to that, we're of course also working on substantiating the SynList more within the groups that we have already. So we say that we have 3 neurotoxicants on the SynList now. If we get access to data, making it possible to identify yet more chemicals, for example, in this group or in other groups, we will work on that to the extent that our resources allow us to do so. And another strand of work is, of course, that we are always concerned about regrettable substitution. So we are working with groups of chemicals. Hans-Peter has been introducing this idea as well. So for example, we have bisphenol A on the SynList, but also bisphenol S and bisphenol AF, etc. So we're always trying to see, can we strike a balance between not putting too many chemicals on the SynList, because it needs to be a manageable tool, making sure that we prioritize the most important chemicals to phase out, and at the same time not introducing regrettable substitution. So I promise you that we're working on it, but I cannot convey, exactly what it will be.
Thank you, Hanna. I'm sure many people would really like to know that. So with that, I think we should conclude the Q&A, and I just want to say a few words. I mean, I personally think it's truly exciting to have expanded the list, and not only with the persistent substances that are widely used, but also to a whole new hazard category, the neurotoxicants. And the aim is for us, like Hannah already mentioned, not only to advise companies to phase them out, but also to indicate what future legislation, such as the generic risk approach, could be expected to ban, to phase out through legislation. And special credits should be made to the research project 𝘁𝗵𝗶𝗀, funded by EU's Research and Innovation Funding Program Horizon 2020, for making this update possible. Thank you very much. And I also want to express a special thank you to all the people involved in this update, including Agathe and Hans-Peter. So having this high level of scientific evaluation is crucial for the recognition of the SIN list. It is considerable work behind, but like Hannah said, make no mistake, we will continue with new substances, new hazard classes to be added to the SIN list in the future. So thank you very much to everyone that participated in speaking, but also to everyone listening. Hope to see you soon. Have a good day.